Hello students, today is our topic is human excretion. In this regard, we will discuss how human beings maintain their body fluid concentration. Okay, first of all, the body fluid concentration maintained by one pair of kidney. This kidney is looking like this. Here ureter is coming out. This is ureter. This is renal artery, and this one is renal vein. This covering is called capsule. I think you can look easily. This one is capsule. The pore in this site is called hilum. If we cut a section through this kidney, we can see this. This is cortex. Here 85% of nephron is situated. These are called cortical nephron or superficial nephron. These nephrons are a little bit different from juxtamedullary nephron by the length of Henle's loop. Henle's loop is shorter in cortical nephron, longer in medullary nephron. A nephron is 3 to 5 centimeter long, a nephron has two main parts, Malpighian corpuscles and renal tubule. Malpighian corpuscles have two parts, glomerulus and Bowman's capsule, and renal tubule has three parts, PCT, that means proximal convoluted tubule, Henle's loop, distal convoluted tubule, and the end part is connected with a collecting tubule where 5 to 7 renal tubule come in contact to form duct of Bellini. This orange part is called renal pyramid, which is situated in the medullary part of the nephron. Medullary part. Here, collecting tubules are much more prominent. This yellow colored part is called pelvis, from which ureter is coming out. Rather, ureter is originated from that yellowish part. This is ureter. This is ureter. This one is renal artery, and this one is renal vein coming out. That's why the arrow is towards outside, and this was come in inside the kidney. Okay. This blue part is called renal column or Bartini's column. Is called Bartini's column. Okay, we have completed the structure of kidney. I will repeat. A kidney is a bean-shaped organ situated in the abdominal cavity. Situated in the abdominal cavity, attached with the peritoneum. Okay, the fibrous part is capsule, renal capsule, which is outer part. The kidney has two distinct parts. One is cortex and another one is medulla. One kidney has 10 lakhs nephron. 85% of that is situated in the cortical region, and 15% is situated in the medullary part. Each nephron is covered by a tuft of capillaries, so that this cortical part is much red than medullary part, where 15% nephron is there, so that due to lack of some capillary, its color is faint. Okay, and I told you earlier, the length of the Henle's loop is shorter in cortical nephron, longer in juxtamedullary nephron. Okay, that's why reabsorption of water occur much more efficiently in the juxtamedullary nephron. Okay, this is the yellowish part is I told you pelvis, from where ureter is coming out. Okay, this one is renal artery, and this one is renal vein, which carry our essential material from kidney to the circulation of our body. This is the structure of kidney. And I told you that the structural and functional part of kidney is nephron. Each nephron is two part. One is Malpighian corpuscles, one is Malpighian corpuscles, and number two is renal tubules. Renal tubules has three prominent part is PCT, Henle's loop, and DCT. Another part is collecting tubule, which is not included under nephron. The length of the PCT is 20 millimeter and the diameter is 50 micron. Okay, the length of the Henle's loop is 14 to 26 millimeter and the diameter is near about 15 to 30 micron. The length of the distal convoluted tubule is 5 millimeter and diameter is 22 to 50 micron. The length of the collecting tubule is 20 millimeter and the diameter is 40 to 200 micron. These are brass border cell of PCT. These are canaliculi. You can see these are mitochondria. This one is nucleus. This one is epithelial tissue. This one is very specialized structure, is called brass border. Henle's loop is the number of microvilli is very fewer in Henle's loop. It has no brass border. The structure of DCT is very much looking like PCT and it has also devoid of any brass border. Only few microvilli are there. These are nucleus. These are epithelial tissues. Okay. If we try to draw a nephron, you will understand very easily. This coiled part is PCT. From this measurement you can understand this one PCT, the first part of the renal tube, the length of it is 20 millimeter and the DCT is 4 times less. So it is PCT is much more coiled than DCT. PCT ends in Henle's loop. And passing in between afferent and efferent arteriole. This one is glomerulus. I will label it up. Now I have completed the structure of a nephron. This one is glomerulus. These are the podocytes of Bowman's capsule, which is situated in the visceral layer of it, and these are the parietal layer. In between podocytes, filtration pores are there. Here a specialized structure is formed where the afferent arteriole, this one is afferent arteriole, when comes in contact with the distal convoluted tubule, the structure of afferent tubule is changed to form juxtaglomerular cells, and the cells of the distal convoluted tubule is formed macula densa. And in between afferent and efferent arteriole, these are this is efferent arteriole. A specialized structure is called Lassis cells or Pulkysian cell. This one is Lassis cell. I think you understand. Altogether, the juxtaglomerular cells, the macula densa, the Lassis cells, all together is called JGA apparatus. That is JGA apparatus. Do you know the function of JG apparatus? I think you know. From JG apparatus, an enzyme is secreted, is called renin. The spelling is renin, not RENNIN, which is related with the digestion in calf's stomach. This is renin. This renin is responsible for maintaining body fluid concentration. I will tell you. First of all, I want to clarify the structure of nephron. I think you understand all the parts. These are towards duct of Bellini. Towards duct of Bellini. Bellini ends up in this tapering end of the renal pyramid, where 25 nanometer renal papilla is situated. This duct of Bellini ends here, and urine comes out from the renal papilla in the pelvis, and pelvis secreted it to the urinary bladder. Next. I am telling you the function of JG apparatus. Very interesting. From JG apparatus, that is kidney, from where renin is secreted. When our body fluid concentration is lesser, then from the liver which is secreted angiotensinogen. Angiotensinogen compel to produce ACE, that is angiotensin converting enzyme, which convert angiotensinogen to angiotensin 1. When renin comes in contact with angiotensinogen 1, it will convert it to angiotensin 2, which acts over artery and vasoconstriction occur. Not only that, vasoconstriction occur means the blood pressure is increased. Before that, blood pressure decreases by the ample water is there. Vasoconstriction occur, blood hypertension is managed. Another work of angiotensin 2 is to produce aldosterone from posterior pituitary. This aldosterone helps this part is DCT, and this one is collecting tubule, from where sodium ion absorbed and coming out to the body fluid. So the concentration of the blood increases. So in these two ways, first of all, vasoconstriction, that is the contraction of the artery, and second one is production of aldosterone, which helps to absorb sodium ion from DCT and CT to the body fluid. So the concentration of blood increases, blood pressure is maintained. When the thickness of the blood increases, in this way thickness of the blood increases. Before that, thickness of the blood decreased. To maintain the equilibrium, here sodium ion added with the blood and vasoconstriction occurs, so the blood concentration is increased. When it is upper, that is normal blood pressure is 80 by 120. This is systolic pressure and this is diastolic pressure. When it is higher than that, what will happen? Osmolarity increases. What increases? Osmolarity. Osmolarity increases. Osmoreceptor contracts. Osmoreceptor discharge occur, which stimulates neurotransmitters. Neurotransmitter influence supraoptic nucleus of hypothalamus, which induces posterior pituitary to produce ADH. ADH is responsible for water absorption. Here the blood capillaries. If water absorbed by the nephron, that is ascending part of the Henle's loop, the body concentration will decrease. Osmolarity increases, the thickness of blood increases. Osmoreceptor contracts, osmoreceptor discharge occur, which stimulates neurotransmitter, influence supraoptic nucleus of hypothalamus, so which induces posterior pituitary, ADH secreted, water reabsorbed, which is called diuresis. So concentrated urine produced. So again, body fluid concentration is lower than the previous. Next one is counter current multiplier. The third mechanism of maintaining concentration of body fluid is counter current multiplier, which is held in the Henle's loop. These are the descending part and these are the ascending part. This is cortex and this is medulla. Here the osmolarity is 300. Here water is reabsorbed, so the concentration is increases, water is reabsorbed and little bit sodium ion also reabsorbed. 300, 600, 900, 1200. This part, actually ascending part is impermeable to the water molecule, but sodium ion is reabsorbed, chloride ion is reabsorbed, little bit potassium is reabsorbed, so the concentration is gradually decreases. Water is acquired here, 1200, 1000, 700, 400, 100. These are medullary part. Here is 1200, 900, 600, 300. Here concentration is gradually increases by the reabsorption of water. Here the osmolarity is decreases by the reabsorption of these materials, sodium chloride and potassium. So the concentration is decreases here. Water is restored here due to impermeability. So a counter current is observed here. Why it is multiplier? When the concentration of the descending part is increases due to loss of water, the concentration of the medulla is very much alike of the descending part. So a positive feedback, water is coming out here by the descending part, but the concentration of the medulla is not increased. Concentration of medulla is not decreased due to presence of water. This water absorbed by the vasa recta, which is a blood capillary in the Henle's loop, in the medullary nephron, not in the cortical nephron. Absorbed by the, so the dynamicity is restored here. Always moving, that is flow of water, flow of blood is always occur inside it. Here the ascending part, sodium potassium pump is performed here. So always sodium and potassium coming out here. The dynamicity of the vasa recta is inhibited. The sodium and potassium pump is not functioning properly. So here the concentration is increases, but here concentration is decreases. Why it is increases? Due to reabsorption of water. Why it is decreases? Due to by the help of sodium and potassium pump, these are coming out. So the concentration is gradually decreases. When aldosterone is, when ADH is prevalent, reabsorption takes place, permeability increases, water is absorbed, concentrated urine is coming out from the urinary bladder. These are the counter current. And the third one is ANF. NF means atrial natriuretic factor, which is a peptide hormone which responsible for vasodilation of the artery, and it releases sodium ion through the urine. So diluted urine, so the dilution of the blood is occur, and in this way blood pressure maintained in our body. That's all.